Thank you.
Thanks, Akui. Just give me a second. Cool. Awesome. Thanks for the introduction, Akui. Hi, everyone. Today I'll be talking about a study which we ran recently where we implemented the use of continuous theta-based stimulation, or CTBS, to the DLPFC. And we tried to characterize the brain's response by looking at it at a biochemical level, at a network level, and also at a behavioral level. So before I get into the study, I'd first like to talk briefly about the brain stimulation literature landscape. As most of us are aware, a lot of understanding today of how the brain responds to any form of brain stimulation paradigm generally comes from studies conducted on the primary motor cortex. For example, studies have implemented CTBS to the primary motor cortex and have shown a reduction in motor evoked potential amplitudes, suggesting CTBS to be an inhibitory form of brain stimulation paradigm. These findings are further strengthened when you look at other outcome measures such as reductions in functional connectivity and also increases in GABA concentration at the primary motor cortex. And due to these findings, what we do today is essentially we try and use these similar brain stimulation paradigms on other areas of the brain and try to induce or elicit a similar response. For example, we can apply CTBS to the DLPFC and try and influence working memory task performance or demonstrate a reduction in functional connectivity. However, these findings are often hard to replicate, and there are conflicting results when you read at studies implementing similar brain stimulation paradigms at brain regions outside the primary motor cortex. And this is beautifully demonstrated by a study by Luke and colleagues, where they essentially applied the same brain stimulation paradigm to two different brain regions and saw very different results. And this suggests that we need to properly understand the physiological mechanisms that drive how the brain responds to the brain stimulation paradigms we use today. And this is also particularly important if we're trying to get brain stimulation to a point where we want to influence behavior or cognition. As a result, in this study, the aim we had was we wanted to use a multimodal approach to try and understand how implementing CTBS to the DLPFC would influence brain activity, and that in turn would influence behavior. So to answer that question, we ran a randomized crossover study design and recruited 17 individuals. So essentially, individuals either received the real or sham CTBS in their first session, and then went on to receive the other stimulation block in the second. Within each session, we had a pre and a post time point. At the pre-time point, participants were scanned, and we acquired T1s to get anatomical information. We acquired ASL data. I won't be talking about this because we didn't actually use this for this study. We acquired MR spectroscopy data, which gave us information about GABA concentration. We acquired fMRI during rest to give us an idea about resting state functional connectivity. And we also acquired fMRI during a working memory task performance. And this was to try and understand how CTBS would influence behavior. So once participants completed their pre-time point, they were then randomized into the real or sham. And this was followed by getting participants back onto the scanner, and we acquired similar metrics to the pre-time point. Right, so next I'll walk you through the results that we obtained. So you can use the timeline on the side to keep track of what I'm talking about and what's coming next. So essentially what you have before you is information that displays a GABA concentration acquired from the stimulation site, which is the DLPFC, and also a GABA concentration acquired from the visual cortex, which was our control site. Each individual dot represents a participant, and the violin plots represent data distribution. The black lines within each violin plot represents your 25th, 50th, and 75th percentile from the bottom up. Top row is your real CTBS, and the bottom row is your sham CTBS. What's interesting in this, especially for the DLPFC, is that you see individuals are pretty sparse over here and then sort of accumulate close to around 3. But you see almost a reversal effect where individuals are more concentrated around the 3 mark here and then sort of dispersed after the sham CTBS. This was an interesting observation, but when we ran these data through the stats, nothing really came through. So essentially, we didn't see any change in GABA concentration regardless of site, stimulation condition, or even time point. And just to give you a visual of this spectra that represents the GABA. So the blue traces represents each individual's GABA spectra, or spectra trace, and the red represents the group mean. And these are the traces acquired from your stimulation side on your left and the control side on your right. All right, so next so now that we didn't see anything at the GABA concentration or the biochemical level, we then went on to look at resting state connectivity. So these images here represent resting state connectivity between the whole brain and the stimulation site, which was DLPFC. So essentially, your color bar, red represents less connectivity and yellow represents the more connectivity with the stimulation side. But here's a similar story where regardless of time point or stimulation condition, we didn't really see much happening. We then questioned, okay, maybe what we are seeing is probably not happening at the network level. Maybe it's something more localized. So we did a bit more research and realized papers tend to use what we call a fractional amplitude of low frequency fluctuations, or FAF. So these measures are argued to represent local neuronal activity. So we adopted that measure and acquired FAF measures from the DLPFC, which was our stimulation site, ran it through the ANOVAs, and really didn't see much happening here either. Right, so next we went on to look at functional connectivity during a working memory task performance. And for this study, we used the N-back task, with the 0 back as your control condition and your 2 back as your active condition. So the 0 back's represented by your blue to green color bar, and this 2 back represented by your red to yellow. Similar story, not much happening here either. And lastly, we looked into task performance data. So D prime is a very common metric used in N-back studies to sort of characterize accuracy. And we ran that through the stats. Again, nothing happening here. And we also looked further into reaction time data, which also said there was not much happening. So essentially what we're saying is that when you implement one session of continuous theta-based stimulation, we couldn't see any changes at the biochemical, at the network, or even at the behavior level. And where possible, to ensure that what we were seeing was a true null, we ran Bayesian statistics, and we obtained Bayesian factor scores of roughly around 0.33 or even less, suggesting what we were seeing was actually a true and strong null. However, these interpretations need to be taken with a grain of salt, suggesting that because our sample size is pretty small, roughly around 17 individuals, I believe we probably could have benefited with more individuals enrolled in the study. And lastly, we only ran one session of CTBS. Perhaps multiple CTBS sessions could have led to more cumulative effects, and that probably could have come through with the stats, but that again, it's not a guarantee. So lastly, I'd like to thank all the co-authors who've been very instrumental and helpful and helped me with the study, and also Josh, who's been very instrumental in this study and bringing it to where it is today. And thank you to the Institute who made this study possible. Thanks, ABSS.
Great, thank you so much. Thanks for the clear presentation. I also appreciate the thorough analysis, checking multiple readouts. So also, I encourage attendees to post your question using Wooba Q&A function. Now, I have one question, just a quick question. Can you make any comment on the individual differences? Are there any individuals who showed expected changes?
So by expected changes, do you mean the inhibitory direction? Is that what you mean?
Yes, that's correct, yeah.
So just let me bring my slide back up. Sorry, I'm not sharing my screen, am I? Let me share my screen. Cool, I'll just hide that. Especially for the GABA concentration, you do see some individuals that tend to decrease. So this individual is sort of coming down over here. But again, at the group level, it's not really coming through. And this probably is something we may have seen more clearly if we had a larger sample. So with a smaller sample, every individual, I guess, tends to carry more weight. So if they're going in a certain direction, that will influence the stats more. So perhaps a larger sample will give a more clearer picture of what's actually going on.